Now then, with all of that in place, and all of our other preparations in place, we can look at a typical ADME study. And these are normally very straightforward. It's normally a single dose, single period study. Typically now, in reaction to recent regulatory guidelines in 8 volunteers. And with that, we define mass balance by collecting all the excreta, collecting blood samples across the residency period of the study. We're defining the mass balance, and we're creating samples for profiling and characterization. And as we've touched on, we perform that as a conventional study or as a micro-ADME study, depending on the needs of the molecule, also depending on client preference. There's a growing trend in the industry to talk more about doing these micro-ADME studies and reducing the use of animals. We're seeing that a lot in the discussions we get into. We're fairly agnostic, if I'm honest. We're looking at the needs of the molecule, but if clients particularly want to go down a micro-ADME route, then we can do that. The important thing is always to remember that you do need to correlate that back to your animal data.